Hello again. So again, this is Sir Carl. This is your second lesson for this afternoon. So we'll discuss about taxonomy and basic microbiologic techniques. So we'll just discuss the basic microbiologic techniques and its principle. So to begin with, taxonomy. Taxonomy is a science of classification of living organisms. So it's an area of biologic science comprising three distinct but highly interrelated disciplines. So what are these three disciplines? So you have your classification, nomenclature, and identification. So classification is defined as the arrangement of the organisms into taxonomic groups. So on the basis of similarities or relationships. So this may include the kingdom, the domains, the phyla, and the rest. So we'll discuss this on the next slide. While nomenclature is the assignment of names to various taxa according to international rules and guidelines. And lastly, identification is the process of determining whether an isolate belongs to one of the established named taxa or represent a previously unidentified species. So primarily we'll discuss how to identify microorganisms, particularly bacteria. So classification. Classification is an organization of microorganisms that share similar morphologic, physiologic, and genetic traits into specific group of taxa. So your classification system is hierarchic and consists of the following taxa designation. So it starts with the lowest form. You have the species, then genus, family, order, class, phylum, and kingdom. So you have their subspecies. The subspecies are the subdivided based on phenotypic differences, and you have two types of subspecies. You have the serotype, which is based on serologic differences, and biotype, which is based on biochemical differences. So after subspecies, the basic group or the collection of bacterial strain with common physiologic genetic features are known as species. Okay? So this is species. Ito, species. A group of similar species is known as genus. A group or composed of similar genera is known as your family. And then, composed of similar families are known as order. Composed of similar orders are known as class. And group of similar classes are called as phylum. Phylum, or in some textbooks, you have division. And composed of similar division which has similar DNA and RNA are known as kingdom. So these are your hierarchy of biological classification. Okay, so for an example, you have there the human being, the E. coli, how do we classify E. coli, and how do we classify Staphylococcus aureus. So, I want you to look at this table. So, for example, your E. coli belongs to kingdom Prokaryote, phylum or division Firmicutes, class Bacilli, order Bacilliales, family Staphylococcaceae, genus Staphylococcus, species Aureus, and if you still have the subspecies, you have Staphylococcus aureus, subspecies Aureus. Okay? So that's it. For E. coli naman, you have their kingdom Prokaryote, phylum Proteobacteriae, class Gamma Proteobacteria, order Enterobacteriales, family Enterobacteriaceae, genus Escherichia, species coli. If you have biotype, you have E. coli O157H7. So that's it. Nomenclature. Nomenclature is the naming of microorganisms according to the established set of rules by the International Code of Nomenclature of Bacteria, or your Bacteriologic Code. The first name is the genus, and the second name of the organism is called specific epithet. The first and the second names together are referred to as the species. So, just a quick facts, epithets are part of taxonomic name identifying a subordinate unit within a genus. So, remember, in writing genus class, it should be capitalized and followed by the specific epithet, which begins with a lower case. Both genus and species should be italicized in print but underlined when written in script. So if you write it in print, it should be italicized, and if you write it in script, it should be underlined. But when bacteria are referred to as group, like Bacillus, Staphylococci, Salmonella, their names are neither capitalized nor underlined. Identification. So identification is the process by which microorganism key features are delineated. It is also a process of discovering and recording traits of organisms so that they may place in overall taxonomic scheme. So what are the three taxonomic scheme? The first scheme is to isolate and distinguish desirable organisms from undesirable organisms. Second is to verify the authenticity of or special properties of culture in a clinical setting. And last, isolate and identify the causative agent of the disease. The latter may lead to the selection of specific treatment directed toward their eradications, a vaccine mitigating their pathology, or a public health measure that prevent their transmission. So, take note that identification scheme are not classification scheme. Identification scheme are not the same as classification scheme, although there may be some superficial similarities. An identification scheme for a group of organisms can be devised only after that a group has been classified. For example, your popular Escherichia coli that cause hemolytic uremic syndrome in infants, which is your E. coli 0157H7. There are hundreds of different strains of E. coli, but only a few are associated with hemolytic uremic syndrome. So this strain, your E. coli 0157H7, may be identified using antibody reactivity with their O and H antigen. So your O antigen is present in your cell wall, the outer cell wall, if you still remember, while your H antigen is present in your flagella. So identification is further classified into two. You have your genetic characteristics and phenotypic characteristics. So genetic characteristics relates to the organism's genetic makeups. It involves detection of the gene or a part of or an RNA product of a specific organism. It may serve as confirmatory method for the presence of the organism. So common examples, you have your base sequencing, DNA or RNA to measure the degree of relatedness of the two organisms. So you also have phenotypic characteristic, which is based on features beyond genetic level. So this includes readily observable characteristics and those characteristics that may require extensive analytic procedures to be detected. So common examples, you have your colony, morphology, staining, nutritional requirements like using of culture media, biochemical, and susceptibility testing. So now let's go to the basic methods of bacterial identification. So you have here the four most common methods in identifying or classifying bacterial organism. So you have the conventional method using culture media cultivation, biochemical tests, and microscopy. You have analytical profile index for nominal taxonomy, immunologic or serologic tests, and molecular method. So for conventional method, let's start with um, culture media cultivation. So culture media, one criterion is the growth on bacteriologic media. In contrast to viruses class and most parasites, many bacterial pathogens can be isolated in a solid agar-containing media. The general cultivation of most bacteria requires the media to be rich in metabolic nutrients. That is why the components of your culture media are agar or agar, which is a solidifying agent, which is added to the culture media in order for them to be hardened. A carbon source, an acid hydrolysate or enzyme added, which is a source of biologic materials like your casein. So, clinical samples class from a normal non-sterile sites like your throat and your colon or your feces, for that matter, contains multiple species of organisms, including potential pathogens and resident microbial flora. Media can be non-selective or selective. So, in order to distinguish whether that is normal flora or pathogenic, we need to use two types of culture medium. First type is your non-selective culture medium. So your non-selective culture media supports the growth of many different bacteria, thus giving numerous types of colonies. So common examples, you have your blood agar and your chocolate agar. It is called blood agar class because of the blood added as supplementary component in order to facilitate fastidious organisms. So when we say fastidious organism, these organisms that grow requires intense component and easily die. So it's called blood because of the supplementary component, which is your 5% ship's blood. Okay, your 5% ship's blood is the most common blood to be used for blood agar plate. On the other hand, it is called chocolate agar because of its color. It's not because that there is chocolate added to the agar or to the medium, but because of the color. Chocolate agar class, it turns chocolate because it is heated, defibrinated blood. So when you heat the blood, di ba, just like the principle of your dinuguan, for Filipinos, your particular with dinuguan. So if you heat the blood, di ba? If you add blood and then you heat that one, it turns out to be brown. So that is why it's color brown. It's color chocolate. Okay? So chocolate agar is commonly used for isolating Neisseria species, okay, and Moraxella species. So chocolate, because these organisms class require X and X and V factor. So which you will be discussing that one later when you go to Haemophilus and Neisseria species, Neisseria topics. For Neisseria, it requires iron because iron is released when you heat or when you destroy red blood cells. So that is why they will grow more on chocolate agar while less on blood agar plate. Another, because of the diversity of the microorganisms class that typically reside in some sample sites, like skin, respiratory tract, intestine, okay, selective media are used to eliminate or to reduce large numbers of irrelevant bacteria in this particular specimen. So, selective medias are media incorporated with antibiotics, dyes, or chemicals to inhibit the growth of other organisms while promoting the growth of desired organism. So common example, you have your McConkey agar, which contains bile salts that selects Enterobacteriaceae, and your Columbia cholesterol acid blood agar, which is commonly used for streptococci and staphylococci isolation, which contains naman colistin and nalidixic acid antibiotics. So you have their common agents, inhibitory agents, sodium aside that inhibits gram-positive over gram-negative. Again, bile salt, as I said, which is the common agent in McConkey agar, which selects gram-negative enteric bacteria and inhibits gram-negative mucosal and mostly gram-positive bacteria. And your colistin and nalidixic acid, which are antibiotics that inhibits the growth of many gram-negative organisms. So you have here picture of McConkey on the top and CNA blood agar below. Conventional method, another culture media, you have your differential media. Your differential media, these medias allow visualization, okay, of metabolic differences between groups of bacteria. So upon culture, some bacteria produce characteristics pigments and others can be differentiated on the basis of their complement of extracellular enzymes. So the activity of these enzymes often can be detected as zones of clearing surrounding colonies grown in the presence of insoluble substrates like zone of hemolysis in the agar containing red blood cell. So for example, you have your blood agar plate which differentiates hemolytic pattern of streptococci. So for streptococci class, you have three hemolytic patterns. You have their letter A, you have the beta hemolytic. Letter B, you have the alpha hemolytic. And C, you have the no hemolysis, or we call it as gamma hemolytic. Beta hemolytic or complete clearing or yellow zone of hemolysis. Your alpha hemolytic is partial clearing or incomplete hemolysis. So these are described to be greenish zone of hemolysis. And letter C, you have there no hemolysis at all. So you will just see a complete grown of organism. So you have there McConkey. McConkey also is another differential type of media. So if you notice class, blood agar can be used as non-selective. It also can be used as differential. While your McConkey can also be used as selective and differential. So, dalawa yung function nila. So, your McConkey differentiates lactose fermenters from a non-lactose fermenters. So, for lactose fermenters, the colony is usually pink in color because of the lactose fermentation. And for the non-lactose fermenter, it's colorless, or sometimes in the textbooks, it's described as white colonies because they do not ferment lactose, which is present in the culture medium as one of the component. So if you want to know more about differential media class, I posted here a link of my Google Drive. If you click this link, it will link you to a PDF file of the discussion about culture media. Or just simply read Chapter 5 of Jawitz of medical laboratory textbooks. So another example of conventional method is you have your biochemical test. So your biochemical test ultimately asserts the presence of characteristics of metabolic functions and can be used to group bacteria into specific taxon. So the most common examples there are carbohydrates breakdown testing, which includes your TSI or triple sugar iron, your KIA or your Kigler iron agar, and your CTA or trypticase agar. So your carbohydrate breakdown agar, it is used to detect or to test the ability of the organism to produce acidic metabolic products either fermentatively or oxidatively from a range of carbohydrates like glucose, sucrose, and lactose. So, usually, This carbohydrate breakdown class is commonly used in identifying gram-negative organisms, particularly Enterobacteriaceae. So this here is a picture of your TSI. So the yellow is meaning the organisms ferments lactose and glucose. While if ever the organism does not ferment, so it will remain as um, orange or pink. The black here portion signifies that the organism also produce sulfur. Okay? Because there is additional ferrosulfate on the culture medium. Another common example of biochemical is your catalase production. So positive reaction is bubbles or effervescence. So catalase production detects the enzymes catalase that catalyzes the conversion of hydrogen peroxide to water and oxygen. So when a colony is placed class in a hydrogen peroxide, there is a liberation of oxygen as gas bubbles that can be seen. The test is particularly useful class for Staphylococci differentiation from Streptococci. So Staphylococcus produce positive results while Streptococcus produce negative results. Coagulase test is also another common test performed in the lab, which is the positive result is clumping. So coagulase, the enzyme coagulase acts with a plasma factor to convert fibrinogen to fibrin clot. So positive, that is why the positive result is clotting formation. So this test is commonly employed for the differentiation of Staphylococcus over the non-pathogenic Staphylococcus species. So here below, positive results, presence of coagulum. Here, presence of clot after the addition of plasma. Usually, we add rabbit's plasma or human plasma. Another common example is your oxidase test. Your oxidase test detects the presence of C component of the cytochrome oxidase complex. The reagent used to change from clear to colored when converted from the reduced to the oxidized state. The oxidase reaction is commonly demonstrated in spot tests, which can be done quickly from an isolated colony. So, positive result is purple. Okay, so there are lists of different biochemical tests in table 3.2 of Jawit's Medical Micro, and I want you to read that one because that might come out during your post test on Monday. So last method for conventional is your microscopy and motility method. So this is the first step in identifying individual microbial specimen grown in culture or even directly from patient specimen. So you have two tests. Your motility. Your motility is commonly done to detect living microorganisms. So you have their wet mount and hanging drop preparation. Wet mount is just for short-term observation. Here, there's an example. You just add the colonies and then add your wet mount medium or your saline for that matter. Your hanging drop preparation is for longer time observation and it's more reliable than your wet mount preparation. So staining. Historically, staining is commonly done to identify microorganisms. So, gram stain, acid fast staining, and structural staining. So, we'll discuss this. Gram stain, as I said, gram stain is the most common key diagnostic tool in the laboratory. And again, this is discovered by Hans Christian Gram, a Danish bacteriologist. Gram stain class, the primarily basis is for structure the presence of structure of cell wall. So again, gram-positive has thicker cell wall, gram-negative has thinner cell wall. So nearly all clinical important bacteria can be detected using gram staining. The only exception are those organisms that exist almost exclusively within the host cell, like your chlamydia, and those organisms that lack cell wall, like your mycoplasma and ureaplasma. So here are the steps of gram staining, how to perform gram staining. So the gram staining procedure begins with the application of a basic dye, crystal violet. A solution of iodine is then applied, which serve as your mordant. All bacteria will be stained at this point in the procedure as color blue, after which the cell are treated with alcohol as decolorizer. The gram-positive cell retains the crystal violet iodine complex because again the pores will shrink, while the gram-negative cell are completely decolorized by the alcohol because of the lipid soluble component, which is the outer membrane. Okay? And leaving now your gram-negative organism as decolorized. So when we add now your counter stain, your safranin, as the last step of gram staining, the decolorized gram-negative cell will take up the contrasting color while the gram-positive cell will appear still purple because of the shrinkage spores. So the safranin cannot enter to the cell wall of the gram-positive organism. However, class, if you apply too much alcohol, there will be tendencies that your gram-positive cell class will be decolorized. Okay? So that is why proper timing is very important in gram staining as well. So these pictures here are examples of gram-positive cocci, gram-negative cocci, gram-positive bacilli, and gram-negative bacilli. So primarily, the gram-positive cocci here is your streptococci. Here, for gram-negative, you have your diplococci. This is a typical smear of a gonococcal urethral discharge. So these are Neisseria gonorrhoeae. For gram-positive bacilli, these are bacillus. And for gram-negative bacilli, these are your common Enterobacteriaceae, like E. coli or Salmonella. So another differential stain is your acid-fast stain. So your acid-fast stain cells retain the basic stain in the presence of acid alcohol. That is why they are called acid-fast. So the non-acid fast cell lose the primary stain when rinsed with acid alcohol, and the counter stain with a different color ang ma-retain. So a smear of cells on the slide is flooded with carbulfoxin and heated on the steam bath. After this, after 5 minutes, decolorization happen with addition of acid alcohol. And finally, a contrasting secondary stain like blue or green counter stain is applied. So acid-fast bacteria like your Microbacteria and your actinomycetes appears in red, others takes the counter stain. So these are image of your acid-fast staining. So the red rods there are your Mycobacteria, and the background is because of the counter stain, the methylene blue. So you have your special stain. You have your endospore stain. So heat is required to drive the stain into the endospore. So this is an example of endospore stain. So the green structure there again are your endospores. Your flagellar stain, which requires mordant to make the flagella wide enough to see. So here is an example of flagellar stain using safranin, but it requires mordant. So you see clearly the flagella. Capsule stain uses basic stain and negative stain. So this is an example of a basic stain. An example of a negative stain class is using India ink or Negrosine stain. So you will see capsule with halo and the background is black. You also have nucleoid stain, your Fuljan staining. So this stains your DNA. This one. So this is just a summary of the type of staining. So for simple stain again, it uses single stain for merely for visualization or morphological and shape arrangement. For differential stain, it uses different contrasting stain separated by a decolorizing agent. So depende. It can be for identification or it can be for visualization of structure. So common, you have your gram stain and acid fast stain for identification, and visualization structure are your special stain. The second criteria or method of identifying microorganism is your analytical profile index or your API. So these are commonly employed in numerical taxonomy or your phonetics or taxometrics. So your API consists of plastic strips and microtubes that contain dehydrated biochemical substrates. These biochemical substrates class are inoculated with a pure culture suspended in a sterile physiologic saline. So almost all cultivatable bacterial groups and more than 550 different species can be identified using this test. The identification system have extensive database of microbial biochemical reactions, and the numerical clusters derived from these tests identify different strains at selected levels of overall similarity on the basis of the frequency which they share the traits. So common examples you have there API 20 for enterics, API 20NE for non-enterics, API 20S for streptococci, API 20C for yeast, and API 20A for anaerobe. So picture here is an example of API 20E. So on top is a positive results after incubation, while below negative results after incubation. So you'll just compare this one. So in reading this one class, there is a table that you will be using as guide, and then after that you'll input that to a computer or to a software, and then it will give you an identification of a particular microorganism. So I think for some medic texts, this is familiar to you. Lastly, you have your immunologic or serologic tests. So the designation of sero simply indicates the use of antibodies, whether it's polyclonal or monoclonal antibodies, that reacts with specific bacterial surface structures such as lipopolysaccharides, flagella, and capsular stain. So the term serotypes, serogroups, and serovars are for all practical purposes identical, and they are used for specificity of these antibodies to subdivide strain of a particular bacterial species. So a serum antibody detection should be collected again, take note, during acute phase and a period of convalescence phase. So you have different immunologic tests. You have their bacterial agglutination. So positive result is visible clumps or agglutination. Bacterial agglutination is commonly used to identify bacteria that are difficult to cultivate in a medium, and it is usually performed by adding antibodies or agglutinins to bacterial suspension that will bind to the surface of the organism. So common examples, you have your Widdel's test, Will-Felix test, and your febrile agglutinin for the diagnosis of leptospirosis. Another example is your particle agglutination test. Still, agglutination is the positive results. So, It uses artificial carriers like latex particles or treated red blood cells, which can be absorbed, test antigen, and react with specific antibody present in patient's serum. So common example, you have your microhemagglutination test for antibodies to Treponema pallidum, and you have also your passive Hev agglutination test for streptococci. Flocculation test. Flocculation test positive is occurrence of microscopic and macroscopic precipitations or flocculation. So it uses a soluble antigen that reacts with the antibody in serum. So common example, you have your Venereal Disease Research Laboratory or VDRL and your rapid plasma reagent test. These two tests are commonly used or commonly employed for the detection of Treponema pallidum or for diagnosing syphilis. Primarily your primary syphilis. Enzyme link immunosorbent assay. Your enzyme link immunosorbent assay is a sensitive and specific method for the detection of antibodies against certain pathogens. So, this method consists of enzyme-bound antibodies with the antibody binding sites free to react with their specific antigen. So, positive results is colored end product. Another serologic test is immunofluorescence assay. Immunofluorescence assay is commonly used for the detection of bacterial and viral antigen. So, in this method, it uses monoclonal or polyclonal antibodies which are attached to a fluorescent dye, that's why it's called fluorescence, that are applied to an antigen, which is present obviously in patient specimen, and that is previously treated with either formalin, acetone, or alcohol. So, positive result, occurrence of fluorescence seen under the microscope. So, one example, um, direct fluorescence assay or antibody test, an indirect fluorescence antibody for the detection of Borrelia treponema and rickettsia. So what is the advantage of immunofluorescence assay is that it can determine the source of the specimen. And lastly, Western blot. Western blot is based on electrophoretic separation of bacterial proteins in different supporting media. So the supporting media are your agarose gel and your polyacrylamide gel. So positive results are band on strips or patterns of antibodies. So Western blot is a confirmatory test for HIV, HIV diagnosis. So picture here is a Weddell's test. Weddell's test is commonly employed for the detection of Salmonella, VDRL test. Okay, so for Treponema pallidum or diagnosing syphilis, enzyme link immunosorbent assay. This is an example picture of enzyme link immunosorbent assay and your fluorescence detection. So this is fluorescent label antibody attached to a Legionella bacilli. So for Western blot, this is an example of how do we perform Western blot. So I will not discuss thoroughly about this since you are not medtech, and it will be a long discussion if we'll discuss this one. This is just a summary of how do we perform Western blot. So picture here, class A and B. These are examples of Western blot results of a positive HIV 1 and 2 patient. So here, let's focus on letter B. So lane 1 for letter B is just a quality control. Lane 1, 2, and 3 are just quality control at different dilution. Lane 4 are for patient, and lane 5 is the negative control, and lane 6 is the positive control. So you'll just compare that one. So if there will be band strips or pattern of antibodies present, then that is positive. So, diba? If you go back to HIV, there are glycoproteins and proteins positive for that one. So, that's how we identify, how do we confirm if the patient is having HIV or not. So using Western blot methods. So in the Philippines, this is usually performed in reference laboratory. So another criteria, you have their molecular method. So your molecular methods can be based either nucleic acid-based detection method or the non-culturable method. So for nucleic acid-based detection methods, since nucleic acids are isolated from humans, bacteria, viruses, and fungi, so that is why this is the main method. It uses mainly DNAs or RNA probing. So it is utilized for purified DNA preparations, bacterial colonies, and specimens like sputa, tissue area, and pus secretion. So common examples of nucleic acid-based detections are your plasmid analysis, southern blot analysis, ribotyping, restriction endonuclease analysis, PCR amplifications, and the rest. So, the main principle of this test class employs DNA probe technology that identifies organism by probing genetic composition like base pairs and sometimes RNA, ribosomal RNA. So you also have your non-culturable methods. You have your PCR-assisted approach using rRNA to identify pathogenic microorganisms in situ. So clinicians have long been aware of the human disease that are associated with visible but cannot be cultured. So scientists are now using PCR-assisted approach to identify these microorganisms. So common diagnostic or diagnosis which employs this type of method is identifying Toperema whipplei, which is the agent of Whipple disease. Whipple disease, by the way, is a rare bacterial infection that most often affects the joints and the digestive system. So, when Whipple disease interferes with normal digestion by impairing breakdowns of foods and hampering your body ability to absorb nutrients such as fats and carbohydrates. So I guess that's it. That are the basic microbiologic techniques and the principles. So these are the references. If you want to read further readings, you can use these textbooks. Again, we mainly uses Jowitz as our textbook, so you can read it in chapter 3 of your Jowitz Microbial uh, Medical Micro textbook. So questions, clarifications, if you have questions again, feel free to drop a message on my email as well as on my Facebook account. So again, thank you and I hope you learned something for the 2 lessons. So thank you so much for your time and see you on the next lesson.